Good evening, everybody, and thank you so much for coming. Today, our team from the New Hampshire Academy of Science will be presenting our conservation research on the endangered local ladycipher orchids of the Cypripedium genus. My name is Grace Lang.
My name is Nysa. My name is Maya Turbeville. My name is Aila.
My name is Kevin Sun. I'm Ian. I'm Alan. My name is Nicholas Palifka.
I'm Kastner Anderson.
My name's Emelia Ajavo.
My name's Ella.
I'm Ethan Krom.
My name is Martina. Dwyer.
And just as a brief heads up, this is our ladycipher tissue culture group, and then the second half of this presentation will be dedicated to the genetics work that we've been conducting, which will be presented by our second group here. So before we dive deeper into our project, I'd like to spend a bit of time providing some background information about why we're doing our project and how it ties into the bigger picture. So we're currently living through what many scientists call the Anthropocene, and this term is signifying that the time period we're currently living through is the first time in history that humans have caused a dramatic change in the world's environment. Scientists also believe that we're already undergoing the sixth mass extinction, and out of the 5 previous mass extinctions, which you can see on this graphic to your right, the most famous and also the most recent resulted in the extinction of the dinosaurs roughly 65 million years ago. So extinction is a normal part of evolution, and under normal circumstances, roughly 10% of life will go extinct every million years. However, as you can see displayed here, historically during mass extinctions, over 75% of life has gone extinct in roughly 2 million years. So the mass extinction we're currently living through is driven primarily by human action, and it's had a particularly devastating effect on biodiversity hotspots. So biodiversity hotspots are these ecosystems of intense biodiversity, such as the Amazon rainforest, and they're often home to hundreds of thousands of species, many of which are endemic, meaning they can't be found anywhere else in the world. And just a couple hundred years ago, these hotspots covered roughly 12% of Earth's landmass, whereas today they cover 1.4%. And this is largely due to our actions, such as deforestation, habitat destruction, and pollution. And so, due to the extremely complicated and entangled relationships within those ecosystems, this has also placed an immediate threat of extinction on many of the species that thrive in these hotspots. Currently, one of the most famous of these species is the panda bear. And other species include the Sumatran tiger and the white rhino. And so if we don't take action now, you can expect these species and many, many more to be extinct completely within the next 30 years. I'd also like to point out how this would impact us economically. So currently, a lot of the crops that we rely on are either threatened themselves or dependent upon pollinators that are threatened with extinction, meaning they'll be tied to the same fate. So currently, one of those most famous ones is the vanilla orchid, which is where vanilla is harvested from, and also coffee. So I know a lot of people really like Starbucks, but if there's no coffee, there's not going to be Starbucks, and Starbucks is going to go extinct if we don't act soon.
Well then.
Okay, so that was all pretty discouraging news. So now I'd like to turn to the more encouraging news about what we're doing to help prevent this. So currently the IUCN's Red List, which is sort of like the official list of endangered species that literature often refers to, it's largely focused upon animals that are threatened with extinction, and in particular mammals and birds. But in reality, this is an incredibly skewed representation considering plants make up the majority of life on Earth. So plants make up roughly 82.4% of Earth's biomass, meaning approximately 82.4% of the mass of all life on Earth. And just as a reference point, animals in total, not just humans, but all animals make up less than 0.5% of Earth's biomass. And so even though, we're largely focused upon us and like other mammals, and stuff, we're really quite insignificant compared to the plants, but we're also causing all of the issues here. And so that's pretty messed up. And of the plants, orchids are one of the largest and most diverse families with over 26,000 species in it. And because they're so sensitive to their environment, orchids are often the first to suffer from either habitat degradation or biodiversity loss, which is why they're classified as sort of the canary in the coal mine or an indicator species. And so there are several factors that tie into this. Two of the main ones are A, orchids don't have an endosperm, meaning they don't have the nutritious part of the fruit that feeds their seeds. So in the wild, a lot of the times orchids need to rely on symbiotic relationships with mycorrhizal fungi. And second, orchids are incredibly pollinator specific, and many species of orchids only have one or two species of pollinators. And those pollinators are now threatened with extinction. So those orchids are naturally tied to the same fate. There are two general types of orchids, terrestrial and epiphytic. The terrestrial orchids grow in the ground, whereas the epiphytes grow in the trees. Terrestrial orchids comprise about a third of the total orchids, but more than half of the orchids going extinct are terrestrial, which is signifying to us that they're going extinct at around twice the rate as epiphytes, which makes them a really useful tool for conservation research, as we're doing at the NHAS. So at the NHAS, studying the lady slipper orchids has been one of our biggest and most long-term projects, beginning in the 1990s. And over time, this has been divided into several subgroups. So the first half of the presentation, like I mentioned earlier, will be largely focused upon tissue culture and how we grow the lady slippers from seed to seedling in the lab and then gradually transplant them to soil. And all of this will be covered in great detail by my teammates over here. And this is leading to our broader goal of creating micro sanctuaries all over the Northeast. The second half of the presentation will be focusing upon the genetics research that we've been conducting, particularly over the past few years. I'd also like to point out that this isn't really covered in our presentation today, but we also study the lady slipper tissue under the microscope, which is known as histology. And these histological insights tie really well into our tissue culture procedures to maximize our results. So now I'm going to turn it over to Nysa, who's going to talk a bit more about the species we study.
So the reason that the NHAS studies endangered lady slippers is because they are decreasing in population quite rapidly. And by using our efforts to conserve these species of endangered terrestrial orchids, we can use them as an example in saving other endangered terrestrial orchids in the world from extinction. In New Hampshire, there are less than 5 places where the showy lady slipper or Cypripedium reginae is thriving, which is why they're considered critically endangered. Our plan is to use garden clubs and conservation organizations to create sanctuaries, with us providing them with the orchids for free. Now, if this approach works, we will be able to use this as a model in saving many other endangered plants in the world from extinction. A little bit specifically about the showy lady slipper is that it often has 2 flowers per plant, and it likes to live in damp areas known as fens. So plants are categorized by their flower structure, which is why the yellow lady slipper or Cypripedium parviflorum has the same basic structure as the other lady slippers, but its sepals and petals vary in color and slightly vary in shape. So here you can see the fused upper sepal. You can see right up there. And then there's one sepal behind the yellow petal. And then as for the petal, you can see the yellow petal in the middle and then the 2 twisted side petals. And a good thing to note specifically about the yellow lady slipper is that its sepals and petals are often twisted or curled. Okay, so out of all of the lady slippers mentioned so far in this presentation, the ram's head lady slipper, or Cypripedium marinum, is by far the most endangered. And it's also the smallest, with a full-grown plant only being about 1 foot tall. Now, this plant, as well as the yellow lady slippers, like to live in mixed hardwood forests. And a good other thing to note is that the flower of the ram's head is about the size of a US quarter for comparison. Okay, now Maya will be talking about our process.
Thank you. This is our entire process. We start by harvesting the actual seeds, inoculating them, growing them in sterile culture, then planting them in the wild. The process in mid-June is when the lady slippers flower and are naturally pollinated by mason bees. Over the next 15 weeks, the flowers will shrivel, the ovules will mature into seeds, and the ovary will ripen into a seed pod like this one. Then, In mid to late September, we will harvest the seed pods and bring them back to the lab. Back in the lab, we will split open the seed pods and carefully scrape out all of the seeds. Each of these dust-sized particles are seeds. If they were magnified 100 times, they would look like this. This translucent layer is called the outer seed coat. Inside the outer seed coat is the inner seed coat. Inside the inner seed coat is the embryo, which will hopefully turn into a pretty orchid.
To simulate the mycorrhizal roll, we use bleach to erode the seed coats. Here, we are mixing a few thousand seeds in a solution of bleach and detergent. This surface sterilizes the seeds and reduces the chances of growing mold and bacteria, which would overtake the cultures and kill the seedlings. The bleach weakens both the inner and outer seed coats of the lady slipper seeds, giving them a better chance of germinating. The outer seed coat is paler and translucent, and there are white patches on the embryo, where the bleach is eaten away at the inner seed coat as well. Next, we pour the bleach solution into a filter to catch the seeds and wash the seeds in sterile tap water to prevent them from getting completely sterilized, which would kill them.
Once the seeds have been sterilized, we inoculate them onto sterile medium. This medium is a nutrient-rich agar, which we place the seedlings onto using an inoculation loop. As you can see, Emelia is doing that in this image. This agar provides all of the nutrients to the seeds that they need to grow because, as Grace mentioned earlier, they don't have an endosperm, meaning that we need to provide it with nutrients. After 3 to 4 months in sterile medium, the seedlings will have grown to look like this. As you can see, there are shoots and roots, and also a rhizome, which is this underground portion where the shoot meets the roots. The rhizome is actually an underground stem. And as you can see here, not all of these seedlings are the same size, since some grow much faster than others. And there's the shoot and the root there, and then in the center you can see the roots.
Okay, the next step is transferring the seedlings from the tube to the jar. And as you can see, this is all done in a sterile hood. This is a photo of me doing transfers. So I'm taking forceps, and I don't know, you can't really see it, but there's a back decelerator. So I'm sticking the forceps in there and sterilizing my forceps, and then letting it cool, and then grabbing seedlings from the tube, and then transferring them into jars so they can have more space to grow. And this is a comparison of a jar of seedlings. This lady slipper seedlings to a US dime. And this seedling's around 2 years old.
Now, before we talk about our recent research, I'm going to give a little background on dormancy because it will provide some context for that research. First of all, the most difficult part of getting lady slippers to grow efficiently, the most difficult part of growing lady slippers efficiently is getting them to germinate quickly. And the reason is because mature lady slipper seeds have evolved a stubborn dormancy. Dormancy is a state of inactivity which protects the embryo from dangerous conditions, like the freezing cold in winter, because once a seed germinates, the growing plant is much more vulnerable to being damaged by its environmental conditions. To prevent this, seeds have 2 different ways of maintaining dormancy. They have a physical barrier and a chemical inhibitor. In terms of the physical barrier, seeds are tightly wrapped by their seed coats, which prevent them from absorbing the water and nutrients that they need to fuel their germination. In terms of the chemical inhibitor, mature seeds are full of the plant hormone abscisic acid, which is abbreviated as ABA, which is the primary driver of dormancy by shutting off metabolism. For a long time, the NHAS thought that the lady slipper's stubborn dormancy was because of their formidable inner seed coat, which you can see in this scanning electron microscope image of the inner seed coat of a showy lady slipper, which was taken by previous NHS students. And you can't see it here because this just shows the outside, but the inner seed coat of lady slippers is really thick, and it's full of hydrophobic compounds, which make it especially impermeable to water and make it very tough and resistant, durable. And now recently we're beginning to think that the seeds are also full of ABA, abscisic acid, which makes them even harder to germinate. In the wild, lady slippers need a very specific set of conditions and a very long time in order to germinate. But in the lab, we have found that soaking them in bleach somehow magically gets them to germinate quickly. And for a long time, we didn't completely understand why. In the past, the NHS believed that bleach's ability to get the seeds to germinate was due to its ability to degrade the inner seed coat. But now recently, we're beginning to study whether or not bleach and other compounds might reduce the chemical inhibition ABA as well.
So we designed an experiment to really figure out how the components of bleach affect the different stages of dormancy. In this experiment, we used bleach. And what is bleach? So the standard bleach, commercially available bleach that we use is composed of sodium hypochlorite, which is the active ingredient. This is the thing that actually sterilizes what you're trying to disinfect. And then it also has NaOH, sodium hydroxide, which is a stabilizing agent. It's not an active ingredient, but it instead helps the bleach not go bad very fast. The sodium hypochlorite is known to firstly disinfect, and also in the context of our seeds, it dissolves the inner seed coat, removing the physical barrier to germination. The NaOH that we have suspected for some time now has been inhibiting the chemical inhibitors, and inhibiting the inhibitors actually promotes germination. So, how did we run our hypothesis? So we set up an experiment with 5 different groups. Our first group was our standard control group. This is what we use for essentially all of our growing systems. It's a solution of 0.5% standard bleach diluted in RO water. Our second group most directly tested our hypothesis. It is NaOH, the stabilizing agent, at 2% of 1 normal. But the stabilizing agent might inhibit the chemical inhibitor, but it doesn't actually do any sterilization, which means if we just used solely NaOH, it would grow a bunch of mold. So we had to disinfect it first with 70% isopropyl alcohol. Our third group tested the inverse of our hypothesis, where we just used the active ingredient in bleach without the stabilizing agent. This would remove the physical barrier only. Our fourth group was a neutral control group. Isopropyl alcohol at 70% works very well as a neutral control group because isopropyl alcohol doesn't dissolve the inner seed coat, and it doesn't, as we suspect, also inhibit the chemical inhibitors. So it's very good at just sterilizing our seeds. While doing research for our experiment, we came across a group of scientists that did research on this topic, but instead for corn stalks. And they found that treating the seeds first with sodium hypochlorite and then post-treating it with 2% of 1 normal of NaOH actually worked a lot better. So we took all these solutions, soaked our seeds in them for 30 minutes, and then we inoculated them using our standard procedure.
To visualize the results of our bleach experiment, we created a scale. And this scale goes from 1 to 5, showing the growth of a seed. At stage 1, it is an ungerminated seed, and at stage 5, it is a seed that has fully germinated with shoots, roots, and root hairs visible. As you can see in this leftmost graph, we have the percent of seeds that showed any growth whatsoever in each of the groups. And in this middle graph, we have the percent of seeds that reach stage 4 at the final observation time. And in the rightmost graph, we have the percent of seeds that reached stage 5 at the end of the observation period. As you can see, commercial bleach actually did really well in this experiment. And interestingly enough, NaOH, which is just the inhibitor, actually did not do very well. And we don't quite know why this is, but when testing the inverse of our hypothesis, the NaOCl, which is just the active ingredient in bleach, it also had really great results. And as a third, the cornstalk method, treating it first with the NaOCl and second with the NaOH, ETOH. So first with the active ingredient and then with the inhibitor did well, but not better than either commercial or just the active.
All right, so this is another experiment that we conducted over the summer at NHAS that had to do with bleach, except as the name suggests, this experiment was conducted on the Cypripedium parviflorum, or the yellow lady slipper. And the goal of this was to determine the optimal exenic seed culture procedure for the yellow lady slipper. And in the wild, the yellow lady slipper is one of the most endangered orchid species in New England. And one of the big reasons for this is because the seeds, as said before, have a very thick inner seed coat, which stops a lot of the nutrients from getting to the embryo. And in nature, the seed relies on a symbiotic relationship with mycorrhizal fungi, which helps to weaken the inner seed coat, letting nutrients pass by. In the lab, we use a 0.5% bleach solution to help partially dissolve the inner seed coat, and that lets the nutrients from the auger get to the embryo. And this experiment was to determine the optimal time the seeds need to be exposed to bleach, because too much time in the seeds exposed to bleach could mean the bleach dissolves through the inner seed coat and kills the embryo, which is not good.
So we have 5 test groups. We have the 30 minutes, the 45 minutes, the 60 minutes, the 75 minutes, and the 90 minutes in bleach. And the reason that we decided to do 5 groups instead of 4, like the previous experiment that happened, I think, in 2016, was because we wanted smaller increments between each of the groups, which led to a higher resolution. So this will make our results more specific. And we've designated, we have 40 test groups, 2 test tubes in total, and we've designated 8 tubes per group.
So our procedure for this experiment is very similar to the Cypripedium reginae or the showy lady slipper procedure that was shown earlier. We just took seeds that were harvested earlier that were Cypripedium parviflorum seeds, and we divided them into 5 groups in little tubes, as you can see up there, each for a different test group. And then we fully submersed all of the seeds with our 0.5% bleach solution and put them on a tube rotator for the listed amounts of time. When the time is up, we take the seeds and in a sterile hood, we would pour the seeds and the bleach all into a filter paper in a funnel over an Erlenmeyer flask and wash them with sterile water 3 times. And this is to ensure that the seeds aren't exposed to bleach after the time is up, which could kill the embryo. And finally, we use a small sterile loop, as seen in this photo, and we pick up 30 to 50 seeds from the filtered paper and deposit them onto the surface of the test tube. Now Nysa will show you guys some images we took of the seeds under microscope of different test groups.
Okay, so here are the images of, as Ella said, the different times in bleach under the microscope. And here, they're all, by the way, at 40 times magnification. So here you can see 30 minutes. It didn't do too much to the embryos because the embryos are still pretty dark, as you can see. So that indicates that it's probably not enough time. Then we have the 60 minutes. And as you can see, the embryo, which is this inner part right here, is getting whiter, which means that the bleach is now starting to dissolve the inner seed coat, which is good. And then here, at 90 minutes, we can see that it's much more white and that it dissolved much more of the inner seed coat, which is good. But again, we don't want it to dissolve. We don't want it to get to the actual inner part of the seed, so that, because if that happens, again, that's going to kill the seed and that's not going to be very good. So, yeah.
Okay, so finally, in conclusion, as of now, we've set down the base by counting the seeds in each test tube and checking for germination, which hasn't happened yet, which is to be expected because even in the very successful Superpedium reginae or showy lady slipper procedure, the earliest germination happens at about 3 weeks, and for the yellow lady slipper, it takes even longer. Usually we like to share the pros, but one con or one small issue we ran into was that we grew a little bit of mold along with our seedlings, but we did a repeat experiment and that one doesn't have any contamination yet.
This is the third experiment, which was performed to determine the effect of cytokinins on ram's head germination. So, like the two previous experiments, you might be able to guess that it was focused on improving germination, but unlike the two previous experiments, it has nothing to do with bleach, and it was performed on the ram's head lady slipper. The ram's head lady slipper of the 3 species that we study is not only the most endangered, but it's also the least studied, and only 2 labs in the world are capable of growing them in sterile culture. And of the 2 labs, the New Hampshire Academy of Science is the only one that is actively working on figuring out the best way to grow them, which shows how important any progress is. And you're also probably wondering, what is cytokinins? Cytokinins are a type of plant hormone which regulate growth and development. And there are a lot of different cytokinins. There are over 200 different cytokinins, and they all have a slightly different effect. But what's important is that they all generally stimulate or promote cell division and cell expansion, which enables water intake. And also they are known to inhibit and counteract the effects of ABA. So hopefully all of this should allow seeds treated with cytokinins to germinate more rapidly and effectively. And that's what this experiment was about. So determining whether or not cytokinins promote germination in ram's head, which cytokinin is most effective, and at what concentration.
In this experiment, 2 groups were tested, one with N6-benzyladenine and one with transient riboside, TCR. And 5 groups were tested in this experiment, each with 8 test tubes and 40 test tubes in total. This is an image of us washing the seeds with sterile water and then collecting them in the syringe at the bottom. And then after we collect it, we use a white paintbrush to spread the seeds onto the petri dishes. And that's how we plant them. And so far we haven't had any germination yet, but yeah, we're looking for results.
So I will be presenting our 4th experiment, the activated charcoal. So first off, what is activated charcoal? So it is not the same charcoal used for grilling. It is a highly specialized form of carbon that has been processed to be incredibly porous. This process begins with a carbon-rich material, which is then heated to extreme temperatures in a low oxygen environment. This initial step, called carbonization, removes water and other compounds. The next step is activation, in which the carbon is treated with chemicals at even higher temperatures, which etch away little parts of the carbon structure. This creates a huge network of microscopic pores. The result is a material with a greatly large internal surface area, which is the powder. Did you know a single gram of activated charcoal can have the surface area of several football fields? It is this immense surface area that gives activated charcoal its power. So why do we use activated charcoal? The primary reason is for detoxification. So when our plants are cut and placed into culture, they release stress compounds and hormones. When these compounds are exposed to the air, they oxidize, and that's what creates the familiar browning and blackening of the roots in the medium. This browning can inhibit growth and kill the plants. So this is where the activated charcoal comes in. It is very good at absorbing the molecules, protecting the plants, and cleaning the medium. Other reasons we use it is for promoting root growth and embryo development. Experiments and how we use them. In the experiments, we tested different amounts of charcoal, grams per liter, through putting it in the medium. As you can see here are the different jars with the different concentrations of charcoal. In most of the experiments, we also try to make sure that there's a control group with no activated charcoal to accurately assess its effects. So this is 0.25, this is 0.5. We did a lot of experiments with 0.5 and then 1 gram. Observations, tracking, and results. Having to do observations for 6 different experiments took a lot of work. What I mean is counting the amount of seedlings and shoots in each jar or box, counting the roots, measuring each of them, and then figuring out what level of browning they're on a scale of 0 to 5. I started these experiments earlier with some other peers like Maya, tracking the growth, and now I still continue to make detailed observations of each of the jars and boxes. And now all this will do the brownness good in the system.
To better quantify the results of these experiments, we've created a 0 through 5 grading system to judge the browning of the seedlings. This is what 0 looks like, which has no browning at all. 1 has very slight browning, 2 has slight browning, and in the next row, 3 has moderate browning. 4 has severe browning, and 5 has very severe browning.
We do not have clear results as for growth, but as for charcoal concentration, lower concentrations of activated charcoal promotes more growth. This image shows the comparison between the 0.5 grams per liter in the control batch. You can see that the seedlings in the activated charcoal seem to be browning less than the ones in the control group. Although I must say, a negative effect of the charcoal is that it is a slight blind spot for browning in the medium. We have tried to numerically evaluate the effect of activated charcoal. However, this was very difficult as our observations are qualitative. In general, there seemed to be an overall trend of activated charcoal helping prevent browning, particularly at lower concentrations as shown in the previous images.
So one of our other variables that we tested was the effect of gold and silver nanoparticles when putting into the orchid growth media. So this diagram here shows how the silver nanoparticles specifically form, but the gold nanoparticles that we also use follow the same process. So due to limitations in nanotechnology to this day, it is kind of unknown how exactly they form, but we have narrowed it down to 2 theories, both of which are possible. But starting where it starts, we have solutions of silver and gold salts in an aqueous solution. And then when reduced, they start clumping together. And then when a chemical called PVA comes in, it surrounds them to form the final nanoparticle. So the only difference between the 2 theories is that when they form and come together, the second theory makes them clump in string-like structures, whilst in the first theory, they kind of grow independently of each other. So when dried and scraped, they form this black, very fine-like powder, which we then pour into the growth media when making it. And so the purpose of these trials was to promote seedling health and inhibit specific fungal growth, because a lot of it doesn't actually help it. And so by the end of it, from our observations, it didn't really have a significant difference on the browning of the tips, but it did in fact inhibit fungal growth. By the end of the trial, showing no fungal growth.
As I'm sure you all know, the winters around here are very cold. And for our orchids, if they begin to grow during these cold winters, the harsh temperatures will kill them. So orchids have evolved to have a dormancy period before they begin to grow. And this means that for an orchid to begin its growth process, it needs to go through a period of cold temperatures. This activates some growth hormones. Now, since we are not doing our experiments outside, we have come up with a way to have a synthetic winter inside our lab. And this involves putting our seedlings, once they are ready for outplanting, this involves putting them in a refrigerator at about 3 degrees Celsius to mimic the cold temperatures outside. Once the orchids have gone through this cold dormancy period, they're ready for outplanting. And we do outplanting at many different areas, like in Warren and, like our friend said in the beginning of this, in some of the nature conservatories around here. These orchids are planted in soil, and after years of trial and error, we have found that this is the best combination of soil to have effective orchid growth. And that's quarter dried sphagnum moss, a quarter garden soil, and half promix mycorrhiza. And it is in this soil that we plant the seedlings.
And this is an image of Grace planting the seedlings in a wooden box. And this is it's planted in the soil mix stated before by Martina. And in this image, the seedlings are planted in a clumped formation, and sometimes we do it in a dispersed formation where we do plant it like one everywhere. And after we put the seedlings in the boxes, it's time for us to put like the boxes in soil. And what we want to do is to keep the soil inside the boxes the same height as the soil around it. And afterwards, we put metal screens on top so the voles don't eat the starch in the roots. And every summer, since we plant the boxes, we count how many seedlings come up each year, and this is an image of some people in the summer counting.
This is the data from Warren, New Hampshire. And so there were 29 boxes of seedlings in May 2025. There were 474 seedlings, 78% survival rate, with 1,258 leaves, 74 plants, 131 flowers, 19 seed pods, 26%. Originally in May 2025, there were 610 seedlings and 0 leaves. So over here are some images. This is in the fen, or in Dr. Fletcher's backyard. We have a beautiful array of lady slippers. So in the beginning of the presentation, Nysa was talking about our restoration initiative. So we're giving away boxes. We give away around 30 boxes at the beginning of this year, and we're hoping to give more. And some of these dots you see are some of where our lady slippers are.
So you'll soon learn about genetics. That's the whole portion of taking different genes in the lady slippers and seeing how different individual lady slippers are related to each other. However, here we also do genomics. Genomics is taking the DNA from one lady slipper and getting the DNA of the whole genome of the lady slipper, much like the Human Genome Project done over 50 years ago. So this whole process relies on getting the DNA from the plant and then trying to figure out what that DNA actually is. However, when we get the DNA from the plant, it doesn't come as a whole strand easily read. So when we extract our DNA, it's a different process than what you will see in the genetics presentation. We had to use liquid nitrogen and other ways to make sure we got these long strands, because those were easier to then put together later, as we'll see. So now we have all our DNA strands, and we want to see what's actually in them. We use this process called Oxford nanopore sequencing, which reads the electrical signal from each one of the bases in the DNA to try to figure out what's actually in it. So as you can imagine, different bases have different electrical charges, and it goes through these small pores in a little disc, and then it reads what exactly that strand of DNA is. And then we have all this data that is just the sequence of all the DNA. Then we have to put it together to get the actual genome. We have about 24 copies that we've read, and we can stitch all these together to try to form a coherent genome. That process is called stitching. Let's use William Shakespeare's classic Hamlet as an example. You have all these sentence fragments from Hamlet, and you see these some regions that overlap. I know that, however, these regions in the actual DNA are many base pairs long, much more than just 4 words. But as you can see, these words overlap, and then we can take these small individual strands, these sentence fragments, and we can line them up so that there's a whole sentence, and that we can coherently read Hamlet. At the beginning, what we effectively did is we took Hamlet and put it through a paper shredder. And now we have all these paper shreds, and we want to put them back all together. However, we use these high-speed idiots, also called computers, at the Center for Quantitative Biology at Dartmouth to put these together. Because there are many thousands of these, and it's not something we can do ourselves. But this finally gets us our whole genome of the lady slipper.
Okay, so before we turn it off to our genetics group, I'd like to just encourage you all to spend a bit of time to think about what you can do in your daily life to help prevent climate change. So the position we're currently in is a result of our cumulative actions and poor decisions over the past few decades. But if we all act now, we can also help prevent and even reverse this effect. So as an example, we all drive cars around. But if everybody in this room just drives 10 miles less per week, then we will each save roughly 208 kilograms of CO2 emissions per year. Now, if 100 million people do that, which is roughly a third the population of the United States, then that saves roughly 230 million tons of CO2 emissions. And that is around a tenth of the weight of the entire human population, and also accounts for about 0.05% of total global CO2 emissions. So my point is that something that might seem very insignificant and unimportant and just really small can have a really big impact when we all do our part to help save the world we live in. And also just stop driving your cars. There are better ways to transportate. So before we move on to our genetics group, we're going to pause for a couple minutes and take any questions that have come up so far. Thank you.
The orchids that you give away, they're like your babies. So do you give them to certain people and in certain environments, they'll survive? I mean, how do you know that, for all the work you've done to plant them and grow them, that who's ever receiving them will take care of them?
Well, so we have like this restoration program, and it consists of both like professional like horticulturists and also just really passionate gardeners who want to, help with our thing. And it's an experiment in progress. So part of it is seeing where lady slippers can thrive the best and what the optimal conditions are. So it is like a continual process. It's not like we're just giving them away, but we also do want to make that connection with the community to spread the word about what we're doing and why it's important. And also, we also have like thousands of seedlings, so yeah.
Guys, can I pile on to that? I was one of the fortunate people to be offered the opportunity to grow some in my own garden in Orford. I'm one of the dots there. And I was given a wonderful presentation by some of the students, very good instructions from the staff. I report in on a regular basis and on what's happening. And I will say to you that I've taken this to the extreme because one day I found a new burrow right next to the box. I thought, okay, we're not going to have any groundhogs here. I went and borrowed a trap. Next morning came out, found an animal, and thought, good, I got it. It was a baby skunk. And I am so dedicated to this process that I'm still trying to get the skunk smell out of the clothes that I wore. That skunk was happily relocated to a better habitat.
How are your orchids doing?
I'll tell you. I've got four. They're doing very, very well. Yeah.
Yes.
At one point you said, well, this had to wait for 4 months or something like that. You didn't have that kind of time. How did you manage that?
Sorry, wait, like 4 months?
Yeah, well, wait 6 months or 4 months for something to happen.
Oh.
And you didn't have the kind of time this summer to wait for that to happen.
Yeah, no, we didn't. So we're like constantly doing experiments. It's not like we do one experiment and stop. We often run experiments and at the same time we do like controls in between. So we always have something running and we also check them like every couple of weeks for progress. And so the experiments we started over the summer because the summer program is like there's a lot more dedicated time. So we decided to try out some new ideas then, and then the tracking of the results is just like a more like sort of laid back process that is more gradual and continues.
And what you're doing now in this after school program is a new project or it's a continuation of what you were doing in the summer?
Well, right now we're mainly focusing on the experiments we did over the summer, collecting the data. Well, right now we're actually mainly preparing for Germany. So we're like collecting all of the data we have accumulated over the years, putting it together, and we've been working on our abstracts and we're gonna start working on our posters and papers. So yeah, there's a lot of that going. Yes.
Where do you do all of your research? It seems as if you use very sophisticated laboratories.
At the New Hampshire Academy of Science lab, it's located in Lyme, New Hampshire. So if you know Crossroads Academy,
Okay.
it's like on the same campus.
Are there any places in Hanover that have lady slippers that you're aware of? Have you gotten any seeds planted here?
I believe we have some at Nathan's Garden. Yes, Nathan's Garden. There was a recent initiative at Nathan's Garden. We planted some, unclear of what's the current status of that. But yes, we did plant some in Nathan's Garden. It's maybe when they spring up for like a couple weeks during the summer in like June-ish. So maybe if you go down there, they might be still alive. And it'd be
No, they're gone.
There's one of the Ray School, isn't it, as well? Yeah. I think I saw one of the Ray School, or maybe not.
I'm not sure.
So we'll go on to the genetics, the time is running. But there are about 10 private plots in the Hanover area in people's gardens that have mini sanctuaries that we've been given over the past 10 years. But the past year that we've had this major initiative. So there are a number of people in the Hanover area now that have a box in their yard, like a deer.
So the second part of this presentation is the genetic analysis of lady slipper orchids. I'm Elizabeth Anderson.
I'm Alana Defoor.
I'm Lucia Santos.
I'm Audrey Boland.
And I'm Elena Simon.
So there are two parts. There's the molecular herbarium, where we collect the samples and extract the DNA. And then there's PCR, where you analyze the DNA. So first, I will be talking about establishing a molecular herbarium at NHS. An herbarium is a collection of dried and labeled plant specimens, and it usually lasts hundreds of years due to the storage of drying and storage process. It provides general information. And a molecular herbarium is a collection of DNA samples extracted from plants. This is used for research and conservation. As you can see here, DNA is a molecule of inheritance, and it's a double helix shape. It looks like a twisted ladder. There's a sugar phosphate backbone with the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. Now we will collect our leaf tips for the extracting and purification of the DNA. Our sampling locations for 2025 were in Stratford Town Forest, Vermont, and Eschwell Bog. Plant clumps. Lady slippers grow in clumps, as you can see in this picture. When we are labeling them, we distinguish the clump number and plant number. As you can see, we have 2 different labels for each sample. We have the long plant ID and short plant ID. In the long plant ID, we have the location, the type, the year it was harvested, the clump, and the plant number. And the short ID is a summary of that information. Collecting samples. Leaves are collected with clean forceps and placed into
pre-labeled and cleaned tubes and transported in styrofoam boxes with dry ice. Leaf samples are stored at -70 degrees Celsius in this freezer, as you can see in the picture, and until they are ready for DNA extraction. Next, Harvey will talk about how we extract and purify the DNA.
Once we have collected all the leaf tips, we must extract and purify the DNA to use in the molecular herbarium. In order to get the DNA, we need to get through a couple layers of things that we do not need. The DNA is located at the center of the cell in the nucleus, and the 3 layers that we must break through are the cell wall, the cell membrane, and the nucleus, nuclear membrane. We use a column membrane, a column purifier, which has a membrane in it, which is labeled right there. Once the DNA has been freed using those, once the DNA has been freed, the broken down layers must be removed. This is done using the column purifier. The DNA is stuck to the membrane of the column purifier, and the rest is washed out of it using a centrifuge. Then we repeat that step several times. After that, the DNA is unstuck from the membrane and washed out, and the DNA is ready for the next step. Quantification.
After we extract and purify the DNA, we quantify the DNA. We use the nanodrop for this. The nanodrop is a spectrophotometer, and it tells us how concentrated the DNA is in our sample and if it is usable. How it works. The liquid is placed in the nanodrop right there in the first. The top part emits light and the bottom part receives the light. The machine calculates the DNA concentration based on how much light was passed through. Here's a breakdown of our data table with yellows from Stratfordtown Forest and Esquimalt Bog. As you can see in the highlighted columns, we have the plant IDs. We also have who they were extracted by and the date they were extracted. Here we have our quantification. We quantify our DNA 3 times and get the average for the most accurate result. We store our tubes with the average concentrations in the freezer. These samples will be used for genetic analysis of lady slipper orchids.
So we're trying to figure out the genetic diversity of the orchid populations throughout New England. Genetic diversity is the difference, how genetically different individuals are from one another within a population. So for example, in a classroom setting, 2 people might have blonde hair and blue eyes, and in this way they are genetically similar. But they can also be different, like one of them could be more susceptible to disease than the other. Orchid populations with higher genetic diversity have more genetic differences among individuals within the population than orchids or populations with low genetic diversity. The way that lady slipper orchids reproduce affects the genetic diversity. Sexual reproduction in lady slippers is when the male and female part of the plants, along with the pollinator, produces seeds and the new flowers grow from. And then in asexual reproduction, the orchids clone themselves. Stems from the rhizomes which is an underground, like roots with the orchids grow, and then the orchid clones grow from that underground stem. Orchids grow in clumps, as you can see in that photo, and we are not sure if the orchids within the clumps are reproducing sexually or asexually. You may be wondering why it would matter if they are reproducing sexually or asexually. And the answer to that question is it affects the genetic diversity. Asexual reproduction indicates low genetic diversity because the orchids are cloning themselves, which means they have the same exact genetic makeup. Sexual reproduction indicates higher genetic diversity. With lower genetic diversity, it is bad for population health because it means that the populations would be more susceptible to going extinct due to disease. This is because if one plant in the population were to be affected by a disease and die, it would be most likely that all the other plants would also die because they share very similar genetic makeup. Orchids plants are likely interbreeding. So that means that the plants all have very, very similar genetic makeups, sort of like siblings having similar genetic makeups. And that this makes finding concrete genetic differences among them very difficult. It would be like trying to find genetic differences between people at a family reunions. So we use microsatellites to look at genetic diversity. Microsatellites are short sequences of repeating DNA of about a couple hundred bases. So like, for example, GGA, GGA, etc. They are non-coding, which means they're not crucial in the formation of proteins, and they do not affect survival or reproduction. And this means that they're more susceptible to mutations over time. Because of these mutations, microsatellites are good ways to see genetic diversity within genetic differences within species that have very similar genetic makeups of plants. We study microsatellites using methods that will be talked about later in this presentation to create phylogenetic trees. Phylogenetic trees are diagrams that act as a visual representation of the way that orchids are similar and the way that they vary from one another. It basically shows the genetic diversity. When thinking about phylogenetic trees, you can think of them like a tree, hence the name. You have the trunk, which is the common ancestor that all of the orchids on the tree share. And then you have the branches, which is common ancestors of only a few of the plants. When reading the phylogenetic tree, the more related plants share the same sequences. So, for example, plant A and B are very genetically related because they share the common ancestor of this one and this one. And then plant D and E are less genetically related because they only share the common ancestor at the trunk of the tree. Next, we'll talk about amplifying sequences using PCR.
In order to make a phylogenetic tree, we need a lot of raw data, which comes from PCR, or amplifying the sequence. This is after we use the nanodrop to quantify the DNA. PCR, or polymerase chain reaction, is a lab technique we use to copy a specific segment of DNA billions of times in a few hours. This prepares the DNA microsatellite for further analysis, like gel electrophoresis. We can then determine how genetically diverse the lady slippers populations are. This is a diagram representing how the amount of DNA increases exponentially throughout each reaction cycle. This is a diagram representing the overall process of PCR with 3 temperature stages: denaturation, annealing, and extension. We run the samples through a thermal cycler, which is a machine that regulates the heat stages and runs the reactions. After the machine has ran many reactions cycles, we're left with billions of copies of the specific microsatellite we're trying to amplify. Once we do PCR, we can visualize our results to see if it was successful through gel electrophoresis.
Okay, our next step in the process is verifying sequences. What is gel electrophoresis? Gel electrophoresis is a technique that we use to separate molecules like DNA based on size, using gel and electric current. We use it to estimate how long our DNA is. Ladder is a DNA marker that tells you the size of the DNA sample that we want to measure. In the first lane, the marker is kind of like a ruler, and it represents DNA from 100 base pairs to about 1500 base pairs. Each band is different by about 100 base pairs. To read it, you have to align your DNA sample to the ladder. For example, this base pair is about 300 base pairs long.
Hispanic beverage. As you can see, the bands that we're looking for are boxed in purple. We know that because this is our expected DNA size. But at the bottom, we don't record these because they're below 100 base pairs. These are called primer dimer. This is due to when primers bind to each other.
Alrighty, so that brings us to our last step in our flowchart, from verifying our sequences to aggregating our data. Okay, so this is our table of sequences present in top gem showies. So on the left side here, you can see all the primers we used, and across you can see the short plant IDs, just stating the location that we gathered the plant and the number plant. Now the bolded columns here and here show that these are different plants, but a part of the same clump that we gathered. So TO2 and TO3 are the different plants, a part of the same clump. Now, the colored boxes show a successfully run PCR group from the band showing up in the gel electrophoresis while using these primers. So our next table, a lot more data. But basically, the different colors show a different teammate working on those sequencing. But basically still shows that a band showed up in gel electrophoresis and that PCR process worked. And these are the sequences present in Stratford. And that brings us to our next data table of the sequences present in Esquibog showies. And now for our phylogenetic trees that were stated earlier, we will then use this data to put in a computer, which will then create the branches to then create our phylogenetic trees in the future. Now that brings us to our acknowledgements. Thank you, Dr. Mass, Dr. Michael, Dr. Faletra, Olivia Straight, Brent, and all of our other mentors, family and friends, everyone who showed up to listen to us today, everyone on this board, and Hal for hosting us today.